Biology review on cell growth and reproduction. The first thing that you should be able to do is to describe the events that occur in the cell cycle. So off to the left, there's a diagram of the cell cycle. Now, when a cell is not actively dividing, this is called interphase. When the cell is actively dividing, we call it the mitotic phase. And the mitotic phase is divided into both mitosis, which is the division of the nucleus, and cytokinesis, which is the division of the cytoplasm of the rest of the cell. In interphase, it's going to start with G1, where the cell is going to grow in size. A lot of normal cell functions are going to be occurring here in G1. If there is a growth factor that's received by cells, what they will have happen at the end of G1 is they're going to transition then to start to reproduce into the S phase. If there are cells that you do not need or want to have go through the cell cycle and to divide, cells can exit the G1 stage and enter into the G0 stage, which is where they are going to stay and go through normal cell functions, but not actively divide. In the S phase, this is going to stand for synthesis. So here, your DNA is going to be copied. After the S phase, and it's been double-checked, the cell is going to enter into the G2 phase, the second gap phase. The cell is going to double check to make sure that not only the chromosomes and DNA are correctly copied, but to make sure that it has an appropriate size, appropriate amount of organelles needed in order to enter into mitosis. If these checkpoints are met, then the cell is going to enter into mitosis, where the cell nucleus is going to divide. And then, of course, after the nucleus divides, then you have cytokinesis happen. So the stages of mitosis. First, you are going to start with prophase. Prophase is going to be where you first start to see the chromosomes appear. So we'll see it in 1 second. You can see these X structures are your chromosomes. Next, metaphase, they line up in the middle. Anaphase, sister chromatids get pulled apart. And then finally, telophase, you reform the nucleus, and those chromosomes also start to uncondense. Finally, you saw that there was cytokinesis, which is not a division of the nucleus, but division of the rest of the cell. So it's part of the mitotic phase, but not a stage of mitosis. Now, in meiosis, there are actually 2 rounds of division. Some important things that happen in meiosis is that with your homologous chromosomes, something called crossing over happens. You can see that they've exchanged pieces of the chromosome with one another. And this is going to result in cells at the very end, after the second round of division, having only one set of chromosomes, and all of these cells being genetically different from one another. So what I would know is that there are 2 rounds of division in meiosis, and the end result are 4 haploid genetically different cells. So if we compare the processes, mitosis, division of the nucleus, but you are going to create clones or identical cells. Whereas in meiosis, you are going to be creating genetically different cells. Also, as you saw, meiosis, you keep the same number of chromosomes, whereas in meiosis, you are going to reduce the number of chromosomes by half. If we start with 46, we'd end up with 23 in meiosis. So, what to write down? For mitosis, it's going to create 2 genetically identical diploid, referring to the fact that if this was our body cell, we would have 2 sets of chromosomes. We got 23 from mom and 23 from dad. And each one of these resulting cells is going to be exactly the same as the one that started. So 2 genetically identical diploid body cells. Somatic cells is another term that means the same thing. Meiosis is going to create 4 genetically different haploid gamete or sex cells. So haploid referring to the fact that there is only 1 set of chromosomes, not 2 like in mitosis. This is important because when 2 sex cells come together, they're both going to bring with them 23 chromosomes, meaning that that zygote or fertilized egg cell will have 46 chromosomes. Now, how do we even make those chromosomes to begin with? Well, you will replicate your DNA. So the process of DNA replication that allows for this transmissions of chromosomes and to conserve the genetic information deals with DNA and how it's replicated. DNA is going to be split, so each strand is going to be used as a template. And notice how you have one old strand that's being read by this enzyme DNA polymerase, and it's going to add on those complementary nucleotides. So if there was an A, it would add a T, or if there was a C, it would add a G. We call this kind of replication semi-conservative. So since we're using a template, it can happen a lot faster. And since we're using a template, it can also act to make sure that we reduce the amount of errors. And therefore, we can conserve our genetic information and not have as many mutations as there would be without semi-conservative replication. One other thing I wanted to mention is when DNA replicates, there is one strand that we call the leading strand that can be replicated continuously, and one strand that's the lagging strand that has to continually backtrack because it goes against the direction of helicase. And so this one is slower to be able to replicate than the leading strand. So how do DNA and chromosomes, how are they related? Well, DNA is going to be found if it's a eukaryotic cell inside the nucleus. DNA by itself is going to be very loose, and in order to make sure that it's packaged up so that it can be delivered correctly to the new cell, it needs to condense itself around histone proteins to form chromosomes. So chromosomes are just clusters of DNA. DNA coils itself up to create chromosomes. How are genes and alleles related? Well, genes are going to be segments of DNA and therefore segments of chromosomes that code for a trait. Now, this could code for a protein or it could code for some other type of regulatory RNA, but it's going to code for a trait. And for your chromosomes, you inherit one from mom and one from dad. These chromosomes that you inherit from each parent, but that code for the same traits, even though they might have different versions of them. So different versions of traits are called alleles. And so therefore you inherit one allele from mom and one allele from dad for each trait. We call these two chromosomes together homologous. Homologous chromosomes code for the same traits, but they can have different alleles on them depending on what your parents gave to you. I hope that helps. Have a great day.